My name is Cara Clark. I'm with the, can everyone hear me? Yeah, okay, great. I'm with the Central Coast Wetlands Group at Moss Landing Marine Laboratories. And I'm going to talk about a project that we've done over the last few years to validate CRAM for depressional wetlands, slope wetlands, and vernal pool wetlands. In its early days, bioassessment was really focused on streams, but in the last several years, it's really moved into other wetland types, particularly depressional wetlands. So that'll be some of the data I'll be showing. So in the process of developing CRAM, there are several phases for each wetland module, so each type of wetland. The first is to define what that wetland type is. What is a slope wetland? The next is to create the design of CRAM. And then the third phase is a verification phase, which we sometimes call the laugh test. Some experts take the module out into the field and they determine if they think it works or if they just laugh at it right away and think it's crazy and ridiculous. And so that's sort of field testing the module, bringing it out onto the ground. And then the fourth phase, which I'm going to be talking about in detail, is the validation. And so this is where we compare CRAM to level 3 data. Things like benthic macroinvertebrates, birds, water quality, etc. So validation has been defined as this process of documenting relationships between CRAM and these independent measures of condition to establish its defensibility and its repeatability as a meaningful measure of wetland condition. So in the process of validation, we have to identify the level 3 data that we can use to assess the efficacy of CRAM. So the ideal data set would be statewide, that covers a range of wetland health. It's a standardized assessment throughout the state, truly indicates a gradient of condition, and ideally is collected at the same time as the CRAM data. So in this talk, I'm going to go through the 3 wetland types and show some of the data that we collected and just a few of the results that we got. So first we're going to talk about depressional wetlands. Depressional wetlands are very diverse. There are a lot of different types of depressional wetlands, including golf course ponds, old stock ponds, managed duck ponds, vineyard detention basins. There's just a really wide range of depressional wetlands, and so we tried to get a breadth of types there. The validation data that we used was an algae index of biotic integrity as well as an invertebrate index of biotic integrity. And we also collected some water quality data. And we were really lucky with this project in that we were able to leverage some existing data. There was some great data in Southern California. SWRCB, headed up by Jeff Brown, had that data. And then the Bay Area, Kevin Lundy had a lot of data on depressional wetlands in the Bay Area. So we were able to use those existing data sets, and then we wanted to expand that range so that it became a statewide comprehensive assessment. So we collected new data in kind of northern and eastern California for a total of 45 sites. We went out to these sites across Northern California and collected water quality with the sonde, algae, and bugs. This is showing the algae collection, which included epiphytic algae, where we scraped the algae off of plants that were in the wetland. We sent those off to a lab, and they identified them. And these are actually showing some of the images of the algae species that were found in our samples. And they're just really amazing to me, these really microscopic images of these intricate diatom algae. We also collected macroinvertebrates and worked with ABL to identify our samples. And so this was the set of sites that we ended up with. We had that sort of cluster in Southern California and the Bay Area, and then we really expanded it through Northern California. So we did our best to get broad geographic coverage. And we found that there was a significant correlation. Looking at the first plot on the left is the CRAM versus Benthic Macroinvertebrates. It was highly significant, had a low p-value. And you know, this is real world data, so it's not gonna look like an exact straight line, but they were significantly correlated. It was also correlated with algae, with the algae IBI, Index of Biotic Integrity. And that one was also highly significant. So we were really happy with that because it really shows that the Depressional CRAM module is measuring some of the same things that the biota are responding to. We also drilled down to the attribute level and looked at correlations with individual CRAM attributes and some of these level 3 measures that we used. So the hydrology attribute, for example, was also correlated with both the algae and the bugs. This is interesting to me, especially because you know, hydrology measures things in CRAM. There's an emphasis on kind of watershed stressors and watershed land use, the area that is draining to the depressional wetlands in this case. And so it shows that the bugs and the algae are really responding to some of those same watershed stressors. We also saw some regional patterns, and this was interesting because it shows that CRAM is sensitive to differences in regional condition. It looks like in this particular study, the sites with the highest scores were in the Bay Area, and so that was sort of the greater Bay Area, included up into Point Reyes and some of the East Bay hills as well. So we're going to move on to the slope module. Slope wetlands, as Sarah mentioned, include several subtypes. Wet meadows, both channeled or non-channeled. Seeps and springs, where groundwater emerges, usually on a hill slope. And forested slope wetlands, which are groundwater-dependent wetlands with trees, and often include a lot of floodplains. So a lot of large floodplains associated with riverine systems fall into the slope category. With slope wetlands, they're often particularly impacted by hydrologic stress. Things like ditching and draining, as well as overgrazing, can cause incision of channels and eventually results in a lowered groundwater table, which is really stressful for those types of wetlands because they rely on groundwater to support them. So when we set up the sites, we tried to find sites that were, you know, in more reference condition, and then other sites that were more impacted by some of these stresses. And the validation data that we used included data from both the Forest Service and the National Park Service. They both had monitoring going in their slope wetlands using pretty similar protocols, and these were vegetation monitoring sites. And this was a great data set to have access to because it had a fair range of condition. There were some sites that were more impacted and obviously, you know, National Park Service, the sites were mostly in Yosemite and Devil's Postpile, so that there were some higher condition sites there. It was a standardized assessment data collection method. The vegetation data does indicate something about the condition of the wetland, and there's a lot of indices based on floristic data. And they were out there collecting during a similar time, during a similar time as when we were planning to do the CRAM assessments. So that was great, but it was limited geographically. It was only on National Forest lands or National Park Service lands. So there really weren't any sites on the coast or in the Central Valley. So we went ahead and collected additional data following the National Park Service protocols at 19 sites beyond the ones that we had gone from Forest Service and Park Service. So we had a total of about 40 sites for this project, or this part of the project. So we were able to increase that geographic scope and have a broader climatic and topographic gradient throughout the state. And this was the data collection method, so we set up these plots, which also had subplots, and so it was a very detailed survey of the vegetation at each site. And so this is a map showing the range of sites that we sampled. There's a pretty nice geographic range. We had sites up on the north coast. We had sites out in the sort of Modoc, Shasta region, several in the Sierras, and then along the coast we had you know, a couple down in the Central Coast, and then we also went down to the San Diego area, east of San Diego, where we had some additional Forest Service sites down there that had data already available. So what do we find? This is showing the range of CRAM scores for the slope wetlands that we assessed. They were pretty normally distributed, so that was good for the statistical analyses. The scores ranged from 56 to 90 and a median of 72. So they were on the higher end of things. There weren't any 44s like the one that Sarah showed in her presentation. And that was, I think, an effect of the access to sites that we had, and a lot of the slope wetlands that are severely impacted are on private land, and so we wouldn't have been able to get access to those. But we did have a pretty good range. And we were able to visit lots of different wetland types and sort of climatic regions and areas that had various structures. So it was really interesting to see the diversity of slope wetlands throughout the state. And when we ran the data, we did find a significant correlation with some of the level 3 metrics that we used. So we looked at the percent native cover, and this was from those detailed vegetation plots, and there was a positive relationship with the CRAM index score. So that was great to see. We also looked at something called the Ratliff vegetation score. And this is an index of vegetation quality. It looks at the composition of the species within your plots and their sort of ecological position. And what it's based on, it's based on sort of a conceptual model of grazing impacts and it was sort of developed, I think, primarily for wet meadows in the Sierras. So sometimes it was a little bit of a stretch to take that index into, say, coastal wetlands that don't necessarily have the same structure, but it seemed to work. So the decreasers are plant species that will decrease in cover, have less cover under a regime of overgrazing. So they're sensitive species. The increasers are ones that expand with overgrazing. So these are hardy and much less sensitive. And then the invaders are ones that weren't there before, and after overgrazing occurs, they may colonize and invade and increase. So a higher score, a higher Ratliff score means better condition, and it's a higher percentage of decreaser species and increaser species actually compared to the overall number and the overall cover. So when we looked at that Ratliff data, we found that there was a significant correlation with the CRAM index score. So that was good to see. It's a nice low p-value. And what else do I want to say about that? One thing that we were a little concerned about with the slope module validation and using vegetation data was the potential for autocorrelation, since CRAM also uses some vegetation metrics. But other assessment validation projects have used vegetation data, so we felt pretty good about moving forward with that. And it was what we had access to. There was not a uniform assessment of slope wetlands across the state available anywhere. So there's a project for someone. Whereas, with the depressional wetlands, there's been this development of IBI for benthic macroinvertebrates, and so we really had access to that robust data source. So thanks to folks in this group and others, that data was available. So finally, I want to move on to vernal pools. Vernal pools are really unique, special wetlands. They're characterized by a shallow restrictive layer, usually clay, but sometimes iron or another substrate that causes shallow groundwater to pond during the rainy season. And then they dry down during the spring. They're found in particular locations throughout the state. They're not really ubiquitous across the state. They're kind of clustered in the Sacramento-San Joaquin Valley. There's some in the Bay Area, the Modoc Plateau, the Santa Rosa Plain, and then in Southern California, both San Diego and Riverside counties have a fair amount of vernal pools. And because of their unique hydrology and structure, they have a lot of endangered plants and animals. Probably because they're seasonal, it's pretty easy to develop over them. And also because they rely on that shallow restricted layer, so when they get impacted by ditching or ripping, then they kind of go away pretty quickly. So they're important, and they've also, CRAM has been incorporated into the regulatory process for looking at vernal pools, which Cliff will be talking about next. So we really wanted to make sure that the module was as robust as possible. So we identified a suite of potential level 3 metrics, level 3 data that we could use. We had vegetation data for a lot of the pools. And with the vernal pools, we weren't able to collect our own data. We didn't have the budget for that, and we knew that there was a lot of data on vernal pools out there. So we were able to leverage that data and compile it from a lot of different sources, which was great because we had access to that data, but it was also challenging because the methods of data collection, the resolution, the geographic scope varied quite a bit among the different sites. But we were able to get plant data for a lot of them, and for some of them we only had species richness, and then for others there was detailed cover data, so we could get other metrics like percent native cover, a diversity index, the evenness index. And similar for invertebrates, a lot of the sites had data on the large brachiopods, the fairy shrimp, and so on. And then some of them had more detailed data on all the invertebrates that were found in the pools. So we subset that data where we had it available. So this is showing the vernal pool sites that we sampled. And as I mentioned, they're kind of clustered, Southern California, Central Valley, around the Bay Area. And we had one, or a couple actually, down in the Central Coast in Fort Hunter Liggett. And in Southern California, we had some at Camp Pendleton. So a lot of times these military bases end up as kind of de facto reserves, particularly for vernal pools. So that was nice to have access to that. And then we had one far up in the northern part of the Central Valley. Actually, there were two up there. So the large branchiopod data did correlate significantly with the index score. These are things like fairy shrimp, clam shrimp, tadpole shrimp, which are among the important endangered species found in vernal pools. And as I mentioned, some sites only sampled those types of species and didn't sample other bugs, basically. So we had the highest N for this analysis. And it was highly significant, so that was great. We also looked at the vegetation data, so the Shannon Plant Diversity Index, which is a mix of, the index sort of combines the species richness as well as the evenness of cover of the various species. And so that actually correlated with the biotic attribute score, which was nice because it shows that the CRAM biotic metrics, the vegetation metrics, are measuring some of the same things that the Shannon Diversity Index indicates. So to wrap this up, CRAM scores were correlated with the independent level 3 data. So that really underlines the scientific rigorousness of CRAM and shows that it reflects ecological patterns that are out there in these wetlands. And it's useful in the context of level 1, 2, 3. We don't want CRAM to replace these level 3 assessments because those are really important. But CRAM does allow you to expand both spatially and temporally your sampling regime. So you can sample a lot more sites, or you can sample more times at any particular site using CRAM than you might be able to do with some of these other methods. So the various CRAM modules, a lot of them have been validated now. We just wrapped up this 3-4 year process to validate the 3 modules that I spoke about. And before that, we had validated the estuarine, the bar-built estuarine coastal lagoon module, and the riverine module. So all of those modules are fully validated. They're available for download from cramwetlands.org. We teach trainings on how to use these modules, and when you do CRAM, a training is required because there's a lot of nuances that it's important to communicate to folks. As Sarah said, it's simple, but it's not easy. Next up for validation, which is very exciting, is the episodic ephemeral stream module. And so we're partnering with SWRCB and some folks from Arizona to fully develop that module and to validate it against level 3 data. It's had a lot of attention. There's a draft module module that's been verified. It's gone through that verification step, and it's been field tested, but now it's going to get fully validated, so that's exciting. I just want to acknowledge all the folks that contributed to this project. It wasn't just myself. There were, first of all, our funding from EPA. Couldn't have done it without that. Sarah Pierce and her colleagues at SFEI were major partners in this effort. For the vernal pool data collection, Volmar helped, as well as Lindsay Tunis and Deborah Sykes. And then the samples were analyzed by ABL and eco-analysts. And then a whole slew of folks helped with providing access to sites as well as sharing their data with us. So really appreciate that access to data, because as we mentioned, we didn't have the budget to collect all of our own data. So we really had to reach out to partners and get additional data. So that was wonderful. So with that, I can take any questions. Got one back here. Yep, it's coming here.
For your depressional wetland validation data set, did that include man-made features in the validation set?
Good question, yeah. We did have some highly managed and modified wetlands, things like duck ponds. So I wouldn't call those necessarily man-made, because the entire Central Valley used to be wetlands and now they've just been sort of plumbed. So we had several of those. We had some stock ponds. We did have at least one kind of stormwater pond. So yeah, CRAM can assess both, completely natural wetlands and ones that are more managed or even constructed or restored. Any other questions?
Well, I had a question. So the first section, when you're doing correlations
There we go.
with the IBIs, that was riverine, right?
No, that was depressional. So there's an IBI, Kevin Lundy developed, and some folks at SWRCB developed the IBI for depressional wetlands.
Okay. Oh, that's interesting.
So yeah, and Jeff Brown helped me actually calculate the IBI, which was great.
Very good.
Yeah. Any other questions? Oh, got one here.
So you shared a lot of good, maybe external validation, sort of internally within the program. Do you guys have a sense of sort of if the same crew went back to a site year on year with just natural variability, or between sampling crews? Have you guys sort of validated anything in that arena?
We have looked at the precision of CRAM among teams when multiple teams assess the same site at the same time. What is the precision level with proper training? And we found that it's 6 points at the index level. So a difference of more than 6 points indicates a true statistically significant difference, and anything less than that might be within the range of precision. We've also looked at CRAM over time, more in the context of restoration monitoring. So looking at a restoration site as it matures through its trajectory of improvement. I do trainings in CRAM, so I return to the same sites every year. So I do have sites where I've monitored them every year, but I haven't really delved into that data, but that would be interesting. And yeah, Sarah has something to add too.
So I also train, and...
Wait, use a mic. or you can just hop up here, yeah.
So I also train and have been back to sites for 10 plus years, and it's been really interesting looking at these sites, especially over the past 5 years with the drought, right? At the index score level, you really don't see a giant change, maybe on the order of 2, 3, 4 points or so. It's when you dive down, for instance, like in the biotic attribute level, where the plants are really adjusting to that drought signal, or maybe sometimes there's management actions, and so you'll see those changes in there.
Thanks. Great. Any other questions?